In this session, we discuss failure investigation, root cause analysis, and CAPA. Now, nobody likes a failure, and in the pharmaceutical context, it means a lot of money involved in a decision that leads to rejection. Failure is costly. And if the batch has found its way out of the door into the market, the consequences can be very severe indeed. So, strictly speaking, failure is not an option. But you can't wish failure away. Inherent, intrinsic to any manufacturing process is failure built in. The risk of rejection is inherent to any manufacturing process, which is why we opt for quality systems. The primary objective of quality systems is to put in place policies backed by robust procedures aimed at minimizing the risk of rejection inherent, intrinsic in any process. Now, how you look at failure depends on your own perspective. Dhirubhai Ambani said every challenge is an opportunity, but Toyota chairman went ahead and said every failure is an opportunity for improvement. There are those who say that success teaches you nothing. In fact, it lulls you into a feeling of false sense of security and complacency. That could be dangerous. But every failure is a wake-up call, telling you that there is room for improvement. It grants you an opportunity to start all over again, this time a little more intelligently. After having attempted 1,000 times and finally succeeded, Thomas Edison was reported to have said that he had not only found the one way to do the job correctly, but had also discovered and learned 999 ways in which not to do the job. If you were to look at others who have carried out investigations in other fields, you have much to learn from their experience. For example, they say disasters don't just occur. They are the result of a chain of critical events. In safety engineering, they hold that accidents don't just occur. They are preceded by a series of warning bells which you have ignored. Your body doesn't just collapse. It gives you a series of warnings trying to tell you to go slow, to go check, but you ignore and then you drop. Failure investigation, there is failure, there is investigation. 2 words. Failure is the misfortune of a chain of critical events. But investigation, if it is to be successful, has to find every link in that chain to be able to piece together what happened. Even if one link in that chain is not retrievable or identifiable or locatable, then that investigation keeps hanging fire. It is inconclusive because you could not find all the links that were needed to establish why that failure occurred. Failure investigation is a specialist job. It's not to be done or attempted by amateurs. And these experts have to have certain basic attributes to make their efforts successful. Now let's see what they are. The failure investigation team, designated personnel with technical and or regulatory knowledge and other appropriate attributes and characteristics that have the responsibility and authority to determine what caused the failure. They should be subject matter experts, high on knowledge and also high on experience. They should have a passion for investigation. They should have an investigative bent of mind. They should have keen powers of observation, deduction and induction. They should have this ability to distinguish noise from signal in the data that is available before them. They should have great amount of composure, equanimity and equipoise. They should have great interpersonal and communication skills to place people under investigation at ease rather than terrorizing them. And they should generally be positive, genial and outgoing as persons. Since failure investigation is such a specialist job, and since you need these specialists with these attributes to carry out any investigation, every organization is well advised to form one permanent team called the FI team. And in that team, you may have representatives from different disciplines: production, process development, R&D, QC, QA, engineering. And you may have more than one representative from a discipline. For example, in QC, somebody who's a specialist in gas chromatographs, another who's a specialist in spectrophotometers. In the case of engineering, a person who's a specialist in HVAC may not be the same person who's a specialist on water systems. So you have a team of experts. Not all members of the team would always be required for every failure investigation. Now, if the failure investigation relates to an aspect where certain members of the team have no role to play, they are not to be present. They are not needed, but they are always ready and available. This team acts as the company's rapid response force. Now, what distinguishes a professional failure investigator from others is the fact that the investigator knows what question to ask and why, when to ask, where to ask, how to ask, and most important, whom to ask. They also have this additional attribute that they know where to look, and more important, where not to look. Whatever data is available invariably is a mixture of the signal that you're looking for and messed up with the noise surrounding it. These experts should be able to sift out the noise and retain and keep looking in the direction the signal is pointing. Otherwise, you'll land up with chasing red herrings and your investigation will proceed nowhere. You should have the ability to ask whether this part of the data, is it really relevant to this investigation or is it to be ignored? That comes from experience. We have all been fans of detective novels, and in any detective novel, the author usually places before you all the data that he places before the investigator in the novel. And we all jump to certain conclusions. And as the pages turn, our opinions change, and the investigator is still at the job till you reach the end of the story and you find something altogether very different from what you thought was the case. This is the same in all failure investigations. Now, in international investigation groups and circles, there's a general belief that the prima facie cause for the mishap, as pointed out on arrival at the scene of the mishap, seldom turns out be the correct one. After detailed analysis, the picture that finally emerges points to a totally different story altogether. Some people were interviewed, and I'm placing before you their findings. Alexander Dunn, Director of Assertivity Properties Limited, in a paper posted on the Maintenance World website, quoted a study which showed when trying to prevent unacceptable events from happening again, 10% of participants immediately sought to place blame. 26% immediately expressed an opinion of the causes and offered an opinion without investigating the problem. And only 20% of participants examined the problem in sufficient detail to be able to identify an effective solution. From these statistics, it's clear that effective problem solving is far from common sense. Indeed, research has reportedly proven that unwanted situations within organizations are about 95% related to process problems and only 5% related to personnel problems. Yet, most organizations spend far more time looking for culprits rather than causes, and because of this misdirected effort, seldom really gain the benefit they could gain from understanding the foundation of the unwanted situation. This is according to Jean Bollinger, who writes on the website systems-thinking.org. Now let me caution you, you don't become a failure investigation expert by listening to one hour video or by reading a few books here or there. Investigation is very much like audit. You must be an apprentice serving as an assistant to a group of veterans who go on audit or who go on inspection or who go on investigation. Serve under them, learn from them. And if you're lucky and you have some real lulus of problems that really defy resolution and you're there at it, watching them at work, and over about 5 or 6 such experiences, you're likely to learn the ropes. And over a period of time, you too will become an expert. But it's a long drawn process. It's experience and only experience that will help you become a good failure investigator. Another point I want to mention is failure investigation team need not always be the same team that executes a CAPA. Failure investigation team's specialty is to find out what happened and why it happened. The CAPA team is supposed to be the one that, knowing what had caused the earlier problem, how to go about effecting a remedy that is permanent so that it doesn't recur again. It is quite possible that the same failure investigation team also executes the CAPA, but it need not always be so. So whenever you're doing failure investigation, always bear this in mind, that you should document whatever you're doing and explain whatever you're doing and justify whatever you're doing in such great detail as if you were not to be there and a CAPA is to be implemented in your absence, the people, the team working on the CAPA have enough information, are armed with enough knowledge based on your efforts to be able to do an effective job on the CAPA. So before we get started on failure investigation and root cause analysis, let's try and understand how they are defined. Failure investigation is the process of understanding the key attributes of a particular failure and identifying the likely causes of failure. Root cause analysis is the process of conducting an analysis to identify the physical, human, and latent causes of a particular undesirable event. Root causes are specific underlying causes. Root causes are those that can reasonably be identified. Root causes are those that are controllable and fixable. Root causes are those where prevention is possible. Failure investigation could be triggered either internally because of a batch failure, because of an OOT or OOS in your stability program, or some out of tolerance or deviation noted in production. The trigger could also come to you from an external source. For example, rejection of your batch by the customer, or a very serious customer complaint or series of such complaints, or an adverse event in the market. Regardless of whether the trigger is internal or external, the regulators today strongly recommend that each organization has one SOP for failure investigation. All others should have a point to which they come and where they refer to the failure investigation SOP. In the past, the tendency has been there is an SOP for handling complaints which carries on into failure investigation and CAPA. There is an SOP for OOS which carries on into failure investigation and CAPA. So you had innumerable number of SOPs all repeating what they felt was the right approach towards failure investigation to resolve that particular issue. And if you were to compare these SOPs, oftentimes you will find that there is no harmony. They are not the same. Failure investigation is a professional job, professional procedure to be done by professionals. So the professionals must have a proper SOP that is in place for any failure investigation, regardless of whether the triggers come from inside or outside. Now, a failure investigation normally follows the following steps. Now let's see, what are those steps? Key steps in failure investigation and root cause analysis. First is the event characterization. This is followed immediately with a situation assessment and interim relief measures, if any. Management is now to be appraised and authorization is to be sought from them in writing for launching a failure investigation. Now, the failure investigation could be in one level or two levels depending on the complexity of the problem. You have a common failure investigation SOP, and then the first step is to define the problem statement. This is followed by data collection, then data analysis and review. And if in this process you're able to identify the root cause, then you submit a report to management. Otherwise, if you find that there's more to this problem than meets the eye, that what you have seen is only the tip of the iceberg and the problem could be far more extensive, then you give the interim report to management and seek permission to escalate this failure investigation to the next level. At the next level, you revise your problem statement based on what you've seen. Now you sit down for causal factor identification based on the data available with you. If you feel you need more data, then you must go and get that additional data if it is available. Now you start on what is called the hypothesis development, or theorizing or speculating on what could have happened. Whatever be that hypothesis, it should be tested, if necessary revised, and finally, whatever you have decided on as the most likely causes, you verify it. And lastly, you prepare what is called the root cause summary. It could be in the form of a report, it could be in the form of a map, it could be in the form of a table. These steps are picturized as a process flow in this particular diagram. We now move to the first step in the process, that is event characterization. The initial step in the process is to clearly define the problem or potential problem. This should include the source of the information. This information is important for the investigation and action plan, for effectiveness evaluation, and communicating the resolution of the problem when it occurs. Next is a detailed explanation of the problem. A description of the problem is written that is concise and complete. The description must contain enough information so that the specific problem can be easily understood. Lastly, documentation of the available evidence that a problem does exist. List the specific information, documents, or data available that demonstrate that the problem does exist. This information will be very important during the investigation into the problem. We now move into the next step, that is immediate assessment of the situation and providing for interim relief measures. The situation must be assessed to determine the extent of investigation, and this evaluation should include potential impact of the problem, the risk to the patient, and finally, remedial action that may be required. What do we mean by potential impact? Determine and document specifically why the problem is a concern and what is the extent of impact. These concerns may include product quality parameters or safety or even consistency. When you talk about risk assessment, based on the result of impact evaluation, the seriousness of the problem is assessed. The level of risk that is associated with the problem will affect the actions that are taken and the priority assigned to the situation and investigation. Now you may need to take remedial action. The potential impact and risk assessment may indicate a need for some immediate action to remedy the situation until a permanent solution can be implemented. In some cases, this remedial action itself may be adequate. If so, the failure investigation can then be closed right here after documenting the rationale for this decision and completing the appropriate follow-up. Now, you may have to quarantine the batch under question immediately. Now, you may have to check other affected batches. You may also have to check other affected products. Now, if it is a batch that has failed, what is the future of the specific batch in question? Since products are in a quarantine state until released or rejected, it may be appropriate to retain this quarantine status, particularly if it is expected that the problem can be resolved quickly. Otherwise, failure to meet specifications or non-compliance with the approved process should result immediately in a reject status until the cause of the event and the future of the product is ascertained. If a quick resolution is not possible, then retain reject status, alert planning that another batch may be required, which should take some urgency and pressure off those attempting to evaluate and resolve the problem. If reprocessing is viable, it must be done according to written and approved instruction, involving production and quality control. At this stage, a report is submitted to management, appraising the situation, appraising them of the situation, and seeking from them a formal written authorization for launching the failure investigation. Now, any failure investigation should be done as per a standard approved SOP. The written procedure for carrying out an investigation into any problem is to be created. This procedure should include the objectives for the action, an investigation strategy, an assignment of responsibility, and the required resources. In the objective, it's a statement of the desired outcomes of the failure investigation. The action will be complete when all aspects of the problem have been studied, understood, and underlying root causes, if any, are identified. As far as the strategy is concerned, a set of specific instructions for determining the contributing and root causes of the problem is written. This procedure directs a comprehensive review of all circumstances related to the problem. Finally, assign responsibilities and gather resources. It is important to assign someone the responsibility for each aspect of the investigation. Any additional resources, whether it is financial, personnel, equipment, should be identified and documented. We begin now the failure investigation in its first phase with a draft problem statement. The approach to a failure investigation is the same for any deviation and goes back to a fundamental approach to scientific inquiry. Document clearly what occurred and when it occurred. So you begin the process with a simple statement of the problem. But do not define the cause in the problem statement. Please keep an open mind. Data collection is the next phase where you interview personnel as soon as possible before memories can get clouded and document these interviews. Gather evidence showing the existence of the problem. Collect documents relating to this issue. Gather as much samples as you can. Collect any amount of additional data as may be relevant for the investigation. Having got the data in front of you, review all relevant records and files. Corroborate events through data and records. Document any and all remedial action that may have been taken in the past. Collect any additional data as may be relevant and any additional samples as may be relevant and any additional interviews as may be relevant. Now when you talk about review of records, review the dates, changes, the relationship to other relevant records, the chronology, the history, the interviews associated and duplicates in other locations, if any. When you look at samples, it may be products, it may be packaging, it may be test results, records, etc. The product review, the sample dissection, and further testing if you're carrying out the chemical testing, the microbial testing, the physical testing, and even functional testing. Now, if after this level of review, it is very easy for the subject matter experts to arrive at a conclusion as to what might have been the cause or causes for the failure, you stop right here. For example, a biotechnologist looking at a lyophilized product may be able to immediately point out to maybe a pH shift or a temperature controller that has gone out. A person just by looking at the white powder with black specks might identify where it has got overcooked or the temperature has gone out of control. Where the discoloration that has taken place would be enough for a subject matter expert to know where to look. So if you're able to close the investigation at this stage, then you make a report to management about your findings and stop here. But if the problem is still defying solution, then you know you will have to go in for a more rigorous investigation, for which you need permission from management. So you submit an interim report to management and shift your investigation to the next level, which is level 2. And at level 2, what all do you do? Let's see. A report prepared for complex investigations containing the level 1 report and a more systematic evaluation of the root cause analysis, as the assignable cause may not be initially readily available. Critical deviations are documented in level 2 investigation reports. You start with the revised problem statement. Problem statement revision would be inspection, testing, and review. Are results consistent with the initial problem statement? Now, updated problem statement could be changing the scope, expanding the scope, or reduce the scope as applicable. But whatever it is, be specific based on observable data. We now have to find the causal factor, and you have two options available to you. One is a brainstorming session for hypothesis development, and the failure investigation tools that you wish to use. You have a range of common tools for analysis, the fishbone diagram or the Ishikawa diagram, which is the most common. But you could also, depending on the situation, opt for FMEA or FMECA, FTA, ETA, HAZOP, HACCP, CCA, etc. FMEA is failure mode and effect analysis. FMECA is failure mode, effects and criticality analysis. FTA is fault-free analysis. HACCP is hazard analysis and critical control points. You could use Pareto charts, you could use Run charts, or any control chart methods. You go in for partitioning of variances using design of experiments, analysis of variance, analysis of covariance, and so on and so forth at a higher level if necessary. You have what is called causal factor charting and establish finally the root cause map. But whatever it is, please, there is no substitute for common sense. Use common sense, use tools when they support a comprehensive analysis, not just because they are available. Don't do failure investigation for the sake of failure investigation. Right-size the investigation or analysis. Be sure not to make the problem more complicated than it really is. Next, please avoid analysis paralysis. Keep on analyzing without coming to any conclusion. Analyze the likely causes first. Use feedback loops to refine problem statements and causal analysis. Most problems have more than one root cause. Don't be afraid to ignore instructions and go out of the way or think out of the box. Use the tools in a way that makes sense for the problem. Tools are generic and may need to be modified to fit your specific situation. Don't be tied up with your procedures. The problem dictates the tools, not the other way around. Don't be afraid to trust your instinct or gut feeling. That's why you are a subject matter expert, even when the data does not initially appear to support it. Go looking for that additional data, get that additional data, and vindicate your contention that your gut feel was right. But by the same token, don't get driven away by gut feel and try to fit the data selectively picked to back your hypothesis. That would be wrong. That is not professional. Fine. Now, if you want to identify the cause and establish a causal relationship for the failure, where do you go? Where do you start? First, you draw on your own knowledge and experience. You're not a subject matter expert for nothing. And this is not the first time that you're facing a failure. You've been here, you may have been in other companies and you have encountered other situations. You have all the collective experiences that you can bring to the table to decide on how things went wrong here. What went wrong? Why did it go wrong? Next, apart from your own internal knowledge and experience, you have a whole group of your peers within the organization, outside the organization. You can talk to all of them and bounce ideas and find out what could be the causal relationship. You could survey literature. Today, in the internet world, all you've got to do is to go to a search engine and type in your question, and a whole wealth of information is made available to you at the click of a mouse. Use that. And if all this doesn't quite add up, then you go to the next level of planning what is called experiments or replicates to test out your hypothesis. Find out what parameter or material characteristic is affecting your process in what way. Now, strictly speaking, when we design a process, we like to design quality into that process. To be able to design quality into that process, we look predominantly at two elements. One is the raw material and in-process material attributes or properties or characteristics. Now, what are they? It could be moisture content, it could be particle size, it could be percentage fines, it could be particle shape, it could be bulk density, it could be tap density, it could be particle structure, it could be residual impurities, specific gravity, color, or opacity. It could be the refractive index or the optical rotation or the viscosity or pH or conductivity. Any one of these characteristics or properties in raw materials or in process materials could adversely impact either your process quality or your product performance or your processing time or processing yield your waste products, any one of these. These are defined as critical material attributes that influence quality or performance. The next element are the process parameters. Being able to identify those process parameters that are critical to quality or critical to performance or critical to the processing time, processing yield, the waste products, or any other non-GMP related issue, but which has a bearing on your business. Now, what are those? Could be pressure, could be temperature, could be length, could be RPM, could be time, could be sound, could be weight, could be volume, could be light, flow, humidity, the current or voltage or power, electrical, the delta P, velocity, the pH, or the conductivity. For example, most people are aware that lyophilized product residual moisture may affect stability. Correlation of pH and phase transient temperature may affect finished product characteristics. Excipient concentration and fill volume may influence physical structure and density. Trace amounts of acids, solvents, or impurities may affect behavior of substance in solution or freezing. For biotechnology products that are to be lyophilized, presence of some peptides and globular proteins may interfere with crystallization of excipients. A peptide may orient itself in either alpha helical or beta pleated sheet configuration, depending on presence and concentration of certain organic solvents used in earlier stages of pre-treatment. Shift in pH may cause polymerization, aggregation, or denaturation due to alterations in desired secondary, tertiary, or even quaternary structures. Then the reagent order and rate of addition could influence morphology, yield, or purity. Reagent ratios could influence particle shape or stereospecificity. Type and speed of agitators could influence particle size distribution. The agitator tip speed could influence particle shape. In API, in a chemical reaction, the rate of addition could influence the buildup of concentration at a particular localized level, which interferes with the process. In the case of formulation, solid dosage form, for example, the norm is first active, then excipients, then color. In the case of liquid orals, for example, you have the active solids, then solid excipients, then liquid excipients, the color, and finally the flavor. Now, if there is a change in the order, there could be a change in the way in which the process behaves or the final product behaves. Now, these are all the things that you take to the table before you start what is called brainstorming. And when you do brainstorming, there are some basic rules to follow. Now, what are they? Do not criticize. Do not rule out causes. Do not assess the probability or the likelihood. Look at all elements of the process. And when in doubt, start with the standard 5 categories of process, environment, materials, human factors, and equipment. Other people refer to it differently. Whatever it is, here is the cause-effect diagram for a bag seal failure, where they look at the process, they look at the environment, they look at the materials, they look at the equipment and the human factors. Now you have the main bone of the fish, and then you have the branches and then sub-branches and sub-sub-branches to see what could trigger what to finally cause the failure in the end product. Here's yet another example of a cause-effect diagram, this time for dosage form uniformity. Here you have pre-blending, drying, raw material addition and tableting on one side, raw material addition, granulation, sizing and blending on the other side, together combined to determine the quality of your dosage form or the uniformity of your dosage form. The Ishikawa fishbone or the cause and effect diagram. There are others who refer to the M's. Now what are the M's? Man, machinery, method, that is the processing method, the process itself. Measurement, that includes the analyst, that includes the analytical method, it includes the gauge or instrument. Machinery, the microenvironment, that is the premises, the HVAC, the buildings, utilities, water systems, gases. And if there is nothing else, they refer to majeur or God, that is others, unknown, maybe any other inputs, all leading to the problem that you are investigating. Now for illustrating the procedure, let's also look at the same Ishikawa fishbone and try to see what all can possibly go wrong. Let's start with raw materials. Raw materials and components, one possibility is inadequate definition or agreement or even understanding of specifications between buyer and vendor. Let me illustrate a situation that pertains to this particular point. An order was received, an unusual order, not a regular production run, where the company had bid for a dietary supplement and got the order. The batch manufacturing record was prepared and issued, and the product was manufactured, tested, approved and released. It failed at the customer end. As soon as information about rejection came to the manufacturing unit, people went into a frenzy. What had gone wrong? They tested everything possible, including the retention and reference samples, and found everything was right. They consulted anybody and everybody they could, and all of them said, I don't think you made any mistake. So they went to the buyer. And the buyer said, please sit down, and ran the test in front of them, and the identity test failed. And the reason was very simple. In the specifications, the buyer had said neomycin. But somewhere at the time of quotation, and then there was some communication gap, and it came into the batch manufacturing record as neomycin amide. Now, neomycin amide might functionally do the same job, but the fact was it was neomycin amide and not neomycin. And the buyer was testing for neomycin, and the amide was interfering, and the identity test was failed. Here's a clear example of people who had not understood the specifications clearly before commencing manufacture. What else can go wrong in raw material? Non-validated production procedures at the supplier's end, resulting in variable quality of the raw material. Different test methodologies used by the supplier and the buyer. Or atypical behavior of materials that otherwise conform to specifications. Now, what does this mean? An API manufacturer supplies virgin material, you have no issues as a formulator. But the day he decides, which he is permitted to as per ICHQ 7, to blend the tailings and then supply that as a composite batch to you, it may assay beautifully, so the quality is not compromised in that sense. But the physico-chemical characteristics may have changed because some of the tailings are 2 years old and some of the tailings are 2 months old, and they've been exposed to differing moisture or environmental conditions or storage conditions that has completely changed the physico-chemical characteristics. So when you as a formulator who is doing a physical processing take up this material after having tested it and approved it, you find that the formulation goes for a 6. Lastly, raw material issued. Now, is this possible? Is it possible for someone to issue raw material? Now, let me tell you what all has happened in the industry. In one case, a batch failed because everybody thought, the person in stores thought it was glycerol monostearate. The people checked the label, it's a glycerol monostearate. The people from production checked it out and said yes, glycerol monostearate. Everybody noted it was glycerol monostearate, but what was inside was glycerol distearate. How did this happen? The same vendor supplies GDS as well as GMS, and both are white amorphous powders. Material arrived on the same day, and both GDS and GMS containers were sampled, tested, and approved. But at the time of affixing the label on the outer carton drum, the wrong labels were affixed on the wrong drums. The GDS label went on the GMS drums, and the GMS label went on the GDS drums. So it was a clear case of mix-up of labeling. And the batches failed. The GMS batches failed, the GDS batches failed, and it took quite a while for the company to find out what was going wrong. There was a company making fever medication that looked at a carton that they had imported from Germany, and it said glycerol. So they took it into their formulation without testing, because they had been buying from the same company for 20 years. And they believe that Germany means great quality, so they don't have to waste their time inspecting something that a German company has certified. So based on their certificate of suitability, they skipped the testing, took it into formulation, and 87 children died in Haiti as recently as 1997, because instead of glycerol, it was polyethylene glycol. In a biotech company which had been buying sodium bicarbonate from a very established, reputed brand for years, they had a problem when the batches started failing. Not one, not two, 5 batches failed, and an investigation was launched. And they found that sodium bicarbonate consignment had been delayed, and the people in production were waiting anxiously to get started. On the day the material arrived, the analyst to carry out the identity test was absent, and there was pressure from production. So the manager in QC picked up whatever had been delivered, looked at the label. The name was sodium bicarbonate. The lot number mentioned corresponded to the same lot number that had been supplied earlier, and which had had no problem at all. Because it was material deemed to be from the same lot of the same batch, the QC manager very confidently released the material. But it turned out to be sodium carbonate instead of sodium bicarbonate. The pH would never come down. It was hovering at 11, whereas it should not have gone beyond 8.2 or 8.3. The very fact that the pH was so very high should have triggered an alarm that the material could be defective. But it took 5 batches and an investigation before this was established. Remember, this is an excipient. It's a buffering agent. In one particular case, sodium chloride was supposed to be weighed and dispensed, instead of which magnesium chloride was dispensed. But luckily, the person in production, as soon as he saw the material and felt the material and smelt the material, he felt there was something wrong over here, alerted his supervisor, who went and investigated and found, contrary to what GMP requires, that when you weigh and dispense one item, you should not have anything else in that area. The contract laborer had been told to bring ever so many drums of ever so many different materials to the weighing station because they had to be weighed and dispensed. So he decided to make his job easy by picking all the drums and putting it on the same trolley and bringing it to the weighing and dispensing booth. And accidentally, the person dipped into the magnesium chloride drum instead of the sodium chloride drum at the time of dispensing. But luckily, the batch did not fail. In one company, a contract laborer was asked by the production supervisor, it was the 3rd shift, the night shift, and was told to go to the production stores and bring a drum of paracetamol. Now this poor contract worker knew to read and write his own native tongue, but he did not know how to read or write English. Nobody accompanied him. So he went over there into that area which is not well lit, it is in one corner, And he looked around over there and said P, 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 and picked up a drum, brought it, and the supervisor was not there. And those others in charge of production took it into production, and the batch failed. On investigation, it was found it was not paracetamol, it was some other material whose name also starts with P. And the worst possible case is where a placebo is taken up into manufacture in place of an active. This has not happened once, this has happened ever so many times in ever so many companies. A Brazilian company making contraceptives used a placebo instead of the active. Whether they did it knowingly or unknowingly, I have no information. The fact is, it went into the market, it stayed in the market, and it became a scandal, headline news, and the FDA went rushing to investigate. A company in India, I'm told, even exported a consignment and had to hastily recall it because they realized that it has gone with a placebo instead of the active. These are instances that I know of where there has been a wrong material altogether introduced into production. Now let's look at the manufacturing, the next M. What can go wrong there? The very first thing is weighing and dispensing. And if you were to do a Pareto chart, you might find raw materials and weighing and dispensing are amongst the highest in terms of the causes for failure of a batch. Now, weighing and dispensing the wrong material we have already discussed. What else can go wrong is non-adherence to SOPs in terms of goof-ups and mix-ups. I have repeatedly mentioned that there was this particular young chemist who was supposed to weigh and charge alumina into a phosphoric acid tank to produce aluminum phosphate gel being used as a filter. The person did not carry his BMR with him. The second person who was supposed to be there was not present. He mentioned everything that has to be done on a bus ticket and folded it and kept it in his pocket. And the BMR, because the weighing scale was only 5 kgs and the amount to be charged was 18 kgs, the BMR clearly said, weigh 5, charge 5, weigh 5, charge 5, weigh 5, charge 5, weigh 3, charge 3. He goes across and weighs 5, charges 5, weighs 5, charges 5, and gets distracted with the telephone call, and then forgets whether he has completed the 5 and thinks he has completed the 5, and then just weighs 3 and charges 3. The pH never comes down, the batch fails. This is sheer carelessness. This is unprofessional behavior, but it happens. There was yet another case, I keep repeating. 11.2 kgs of A was supposed to be weighed and charged. The second person was present and they are discussing and distracted. And the person actually weighs 12.1 kgs and charges and writes down 11.2 kgs. And the batch fails and investigation takes forever till raw material reconciliation is done. Yet another case I've often talked about is in a company where 11 kgs of A was to be charged first and 13 kgs of B was to be charged next. The person charged 13 kgs of A and 11 kgs of B. Batch failed. Now all of us are aware, and I mentioned it earlier in this same presentation, that the rate of addition and the order of addition makes a difference. Now, if the persons do not abide by the norms laid down in the SOP, you are likely to have problems surfacing every now and then, especially if people show this level of carelessness in their commitment to their work. Now we talk about the actual processing. Let's take a case like blending. I mentioned that there are ever so many characteristics or properties in the raw material that could influence your blend. Loss on drying, moisture, percentage fines. And there have been ever so many cases where blending just doesn't take place and you have lumps formed. And this goes on forever. And the failure investigation showed that the critical element like moisture or percentage fines had not been identified as a critical element. And therefore, the supplier had supplied whatever was available with him, which was totally unacceptable for the process. Discovered after a lot of heartbreak and tears. There's another situation where the BMR had mentioned that a certain amount by way of active is supposed to be added for 100% assay. And to make matters worse, it was a slow release drug. No matter what happens, now if you were to add active and have your excipients in place and you take samples, normally these people tend to take 3 samples at 3 time points, 10 samples at 3 time points. So totally you get 30 samples. Now, initially you should have 50% above the 100% assay value and 50% below. So you have a scatter, and slowly as the blending takes place, it becomes more uniform. But if it is really done right, there should be as many points above 100% assay as below 100% assay. The question is, what is the RSD that you have decided to set for your process? Now, here, no matter how many samples they were taking, the mean was turning out to be around 92%, and the data scatter was around 3% RSD, around that 92%, but not at 100%, and it was never coming. Even though it was slow release drug, they decided to sonicate before injection, etc. They tried whatever possible, it was not done. It was ultimately realized that there was a calculation error. The amount of active to excipients was wrongly mentioned as something, whereas it should have been about 10% more than what was indicated. That came to light only after all these problems were faced on the shop floor because of scale up. Talking about scale up, because we have an R&D unit that works at 1 liter and 5 liters, and you have to scale up at 50 kgs and 100 kgs and 500 kgs. These issues keep coming up. There was this blend where magnesium stearate was added, and it was just getting to be so sticky that it was sticking all over the blender interior, and the blend was not taking place. And finally, these people had to resolve that issue by adding talc, which was not originally mentioned in the BMR or the scale-up technology transfer documentation, to resolve this issue of stickiness and adherence to the walls instead of blending. Yet another interesting case was brought to my notice, again with these 3 time points, and the process was in the process of being validated and a blend. And a person comes to me and says, I've got 10 S1, S2, S3, S4 samples, and I've got retention samples R1, R2, R3. R1, R2, R3 are well within 90 to 110. But out of these 10 S1, S2, S3, etc., there are 3 which are outside 90 to 110. So what should I do? I said, what are the values? He says, one is 87, one is 89, and one is 117. So I tried pointing out to him that your blend has not taken place. Your blending process has not been completed. Now, this is a point I'd like to mention to all of you. In blending, the right approach would be not 3 ad hoc points in time, but taking as many points in time as possible, from starting from, say, 3 minutes or 5 minutes, and in intervals, depending. Some of you have samples taken at 5 minutes, 10 minutes, and 15 minutes. Some of you have samples taken at 40 minutes, 50 minutes, and 60 minutes. So it depends on case to case. What you really need is to be able to define beforehand what is the mean that you expect and what is the RSD that you're looking for. So effectively, initially when you take, there will be 50% of the points way up and 50% of them way down. The RSD may be 15% or 20% or more. And as the blend takes place, the RSD comes down, and it comes down to a certain level and stays at that level for a certain period of time, and then classification starts taking place and the RSD starts getting larger. Now, whatever is this particular RSD that you get, which is stable over a window of time, that is your blend window. Today, the regulators are encouraging you not to go by process time ending or time-based processing, but go for process endpoint detection and completion. In other words, if it is a blend, use near-infrared probes at different levels, preset what is the mean and what is the RSD that you're willing to tolerate. As soon as those 10 probes that are there permanently inside your blender sense that the content uniformity or blend uniformity has come to that level where it is within that RSD that you have specified, the process automatically stops. This is the way the regulators would like you to progress in the years ahead when it comes to issues like blending or mixing. So we've discussed production-related errors, weighing and dispensing errors, non-compliance with SOPs vis-a-vis goof-ups, non-compliance with SOPs vis-a-vis mix-ups. There could be unusual incidents during production, hoping that production will let you know about it. But the chances are that they won't let you know about it. Here is the case, where the yield is normally 95% and suddenly becomes 72%. So an investigation started, but nobody is cooperating. Anything unusual? No, nothing unusual. But why is the yield suddenly so low? No answers. Somebody tries to misdirect the investigators by saying, here's a leaking valve, if you like you can put your hand underneath, and it is dripping. So they asked, when did you first notice this? They said about 3 weeks ago. Have you informed engineering? Yeah, they have. We have informed them, they're coming, but they've not yet done it. So it could be because of that. So these people went back to check all the batch manufacturing records in the past 3 weeks, and there had been 3 or 4 batches made. And the yield there, there was no issue. It was still in the range of 95-94%. So this could not be the reason. After a great amount of effort, finally the truth came out. The media that they use in that particular process has to be sterilized in an autoclave. And for steam to go in, the lid is kept open. And underneath there's a valve which is also kept partially open. And to prevent the media from spilling out, there is a porous, semi-permeable membrane. The SOP requires, after this sterilization process, that the valve be closed first before this semi-permeable sealing membrane is removed. But on this particular occasion, on this particular day, while the person who was about to close the valve had not yet fully closed the valve, the other person had removed that particular semi-permeable seal, and a certain amount of media spilled out. And this was the reason why the yield was suddenly 72% from 95%. But this took a lot of effort to get the truth out. Now, it could also be because of expected data variability around a specification limit, especially if the process does not have the necessary process capability ratio. What is process capability ratio? How to measure it? What it should be? What is real process capability ratio? Are all things outside the scope of this lecture, but I have addressed this in my lecture on process validation. But if your process is not capable, then you will have 2 issues. One is, every now and then you will have a type 1 or an alpha error, or a chance positive or a false positive, which leads you to reject an otherwise excellent batch, no different from your earlier batch. If your process capability ratio is not defined and your process is not stable, then your process could drift. And sadly, the sample that has been taken is taken from that part of the process or batch which is assaying perfectly. So you are under the impression everything is okay, and so you release the batch. But actually it is a batch that should have been failed. This is called a type 2 error, or a beta error, or a chance negative, or a false negative, whichever language you wish to use. But these are intrinsic to the measurement process and the sampling process. Now, to make sure that this doesn't happen, you should have a high process capability ratio and a process capability ratio that is centered, the process is centered between the specification lines. As I said, this is discussed in detail in the other video on process validation. So you should be prepared that when you have an uncomfortable result, it could be an alpha error which is asking you to reject a particular batch. But worse, it could be also a beta error by which you have passed the batch and undetected OOS has gone into the market and has created a problem in the market which has triggered this failure investigation. Now we have another interesting thing here called wrong product manufactured. Is it possible? Is it possible that production actually goes through the entire process of manufacturing a totally wrong product? Now, I've already discussed one where the specifications were misunderstood and neomycinamide was included as an element, as a component, whereas neomycin was what was asked for by the buyer. But that's an exceptional case. Now, you people have ever so many products erythromycin X, erythromycin Y, erythromycin Z, etc., etc. Now, here was a case where an erythromycin X was to be manufactured. The batch manufacturing record was issued, cross-checked, approved, released, components were released, and the title was erythromycin X, but all the components inside and the instructions there were all relating to erythromycin Z. And this is an excellent case for people who look but don't see. Nobody saw that there was something wrong. Hey, why are you asking for this? Why are you asking for this? It's supposed to be erythromycin X. This is not erythromycin. What you're talking about is erythromycin Z. No, nobody realized it. It went right through till the QC tested it and rejected the batch. And then failure investigation showed that they had made the wrong product altogether. Now, I'll give you another example of people in the processing environment who've all been blessed with a pair of eyes but don't use them. Here is a company which is US FDA approved, MHRA approved, everybody approved, and the multinationals have sent their auditors and they have approved. And there's a large multinational for whom these people make two products. Both are parenterals, aseptically processed. One is an antibiotic and one is a vitamin. To distinguish the antibiotic from the vitamin, the label is red for the antibiotic and the cap, aluminium cap, is red. In the other case, for vitamin, the label is white and the cap is white. A batch starts at 3 o'clock. At 4:30 QC takes samples, at 6 o'clock QC takes samples, at 7:30 QC takes samples. Everybody around the batch manufacturing record has been issued by somebody, has been checked by somebody, has been approved by somebody, and people are dispensing the components. Nobody saw. Till it reached visual inspection where one girl who was a contract worker called the supervisor and pointed out that this is supposed to be a vitamin. Why is the aluminium seal cap red? Everybody was present, but this happened. Batch had to be rejected. A similar situation where a batch starts at 3 o'clock, samples are taken at 4:30, 6 and at 7:30, and it goes past right through to packaging department, and there a contract person calls the supervisor and says, according to the batch packaging record, this is supposed to be the batch number. Can you see what is printed over here? And that was totally wrong. They didn't match. People who look but don't see. How can you explain a situation where a material goes into the market and has to be hastily recalled because on the same strip of tablet, half of that strip says the product is X and the other half is saying the product is Y. Now what product is it? Is it X or is it Y or is it something else? How can something like this possibly happen? I have no answer. But this has happened and a recall has taken place. Now lastly, we have packaging and labeling related errors. We have had a case where a multinational company made hypoglycemic drugs and hypotensive drugs and put all the hypotensive drugs into the hypoglycemic pack and all the hypoglycemic tablets into the hypotensive pack, and people died in the market. A case of mix-up in the packaging department. Yet another case is where everything is right, put into cartons, and outside the cartons labels have to be affixed mentioning the customer's name and address and where the consignment has to go. And 2 sets of cartons are awaiting labeling, and the labels are issued to a contract laborer. And nobody comes along with the contract laborer who cannot read English, who cannot write English, but he has been given the job to go and affix these labels. And this person, because there were identical number of cartons on one side and another identical number on the other side, so he went and affixed the labels. So consignment that was supposed to go to Mumbai went to Brazil, and the consignment that was supposed to go to Brazil went to Mumbai. Now these are all kind of carelessness that have happened, and this is a classic example of a packaging label mix-up. Apart from the glycerol monostearate and diastearate that I talked about earlier. Here's yet another case of a labeling mix-up. Now, while talking about errors in packaging and labeling, one more instance comes to mind where, again, a supervisor in the packaging department told a contract worker to please go to the production department and bring those drums with white tablets, that many drums, 6 drums, 5 drums, whatever, white tablets. And this person went there and got it. When he brought it here, the packaging supervisor was not present, he'd gone away, and people took it into packaging. Halfway through the packaging, a lady operator in the packaging line called the supervisor and said, hey, listen, can you just smell this tablet? Seems odd. This is not supposed to be the way this particular tablet is supposed to smell. So the supervisor smelled it and immediately reacted and called that contract worker and said, where did you get this from? And he said, I'll show you. So he took him there and showed him. He said, no, but not these drums. I asked you to get those drums, those 5 or 6 drums that are there. They are also white tablets. Why did you do this? Why did he do this? Because nobody went with him. He cannot read English. He cannot write English. Nobody escorted him. No responsible person came with him. There is supposed to be a proper issue by the production department and a receipt by the packaging department. Those protocols are all in place, all of which are filled up 15 minutes before the inspector comes. But on the ground, the practice is anybody goes anywhere, picks up anything and starts. And this has happened. Now, what is the next issue we are looking at? Machinery. Now, what can go wrong with machinery leading to a failure? Malfunction of equipment, substitution of equipment, loss of parametric control in the equipment, or part failure. Here was a company that was totally foxed by the total lack of content uniformity or blend uniformity. And nobody was cooperating. In fact, when they looked at the batch manufacturing record, everything seemed to be perfect. When somebody from QA went into the production department to cross-check to see, listen, I just like to see the blender in which it has worked, the person from the production department, the supervisor, got a little aggressive and said, what do you mean? Are you doubting my integrity or something of that sort? So the QA person went and reported the matter to his boss, who talked to the production boss, and all of them together went down. And what did they find? On the batch manufacturing record, it says the blender used was a 300 kg blender because the batch size is of that nature. It needed that blender. But what had actually happened was that that particular blender still had some material from an earlier processing which had not yet been discharged. And so it was not really available. But the pressure to produce was very much on. They had another blender which was the 150 kg blender. So they decided that they would do it in 2 or 3 lots on the 150 kg blender. But this was not reflected in the BMR. The person had packed the 150 kg so tight, and naturally when they were blending, there was no movement of anything. Everything remained solid packed. And at the end of it all, whatever you took out would assay anything but what was supposed to be assayed. This was a case that has actually happened. But it didn't. The failure investigation was not concluded in half an hour. It took quite some time before they could zone in on what exactly was the problem. And the fact that the production was not cooperating didn't help matters any bit. Here we look at microenvironment related errors. Problems could be through seepage through walls or flooring or ceiling, or it could be because of your HVAC malfunction, delta P not being maintained, quality of air not being maintained, air flow not being maintained. Or it could be problem with the water system, a malfunction in the water system. It could be contamination introduced through compressed air or gases or steam. Or worse, it could be power failure together with all related issues that power failure triggers. In this connection, there was one particular company which was breaking its head over its water system. The water quality was simply not up to the mark as far as the process was concerned, and an external expert was called in to find out what exactly is the issue. Looking at the water system, he said, listen, the design is wrong. This water system, it doesn't have the capability to deliver the specifications you're looking for. The mixed bed system is not adequate. In fact, What you really need is an RO system with an EDI unit. And for good measure, add an ultrafiltration stage if you like. But otherwise, you're not likely to meet your specifications. The users were not aware of this, and they were wasting their time trying to figure out what was wrong with their water system. And finally, there could be issues in the laboratory. It could be a sampling error, it could be a sample preparation error, it could be some mistake with the reagents or standards that are used. It could be the equipment malfunction or the instrument malfunction. For example, the column temperature is not being maintained or monitored correctly. Inadequate training, qualification of analyst. There could be an analyst error, it could be a calculation error, it could be a result interpretation error. Or the method itself is not validated or not fully validated. And then you have the measurement system analysis, the whole subject of analyst bias, gauge error, measurement error. And as I pointed out earlier, if your process capability ratio is not where it should be and not positioned where it should be, and if it's not stable, then you could be having type 1 error or type 2 error, and that could lead either to unfair rejection of your batch, and your failure investigation gets nowhere because nothing is different from this batch as compared to any other earlier batches, and it's difficult to find the assignable cause. Or worse, it could be a type 2 error or a beta error which releases a consignment into the market, and the whole consignment represents undetected OOS, which could place your company at risk, it could place your patients at risk. Now, so far, what have we discussed? The Ishikawa fishbone. We've looked at the contribution from men or manpower, from materials, from the manufacturing, from the machinery, from the measurements, from the microenvironment. Now, failure investigation is seldom easy, especially if there is non-cooperation from certain sections affected by the failure. I'll give an example of a particular group that was investigating a batch failure. It was a solid dosage form and the assay was less than 70%. No matter what the investigators were looking for, they could come up with nothing concrete. Everything seemed to be as normal as ever. And this went on for days. Finally, the plant manager got very impatient and said that no one shall ever go home until this problem is resolved. Now, that kind of pressure is certainly not going to help. It only worsens the situation. In this particular case, the people were all sitting by, very dejected, not making any progress whatsoever. And they were sipping tea and it was close to midnight, and suddenly one person said, asked the person, representative from raw material stores, who was also part of the investigation, as to whether any of that material is still left. Person from production said whatever was weighed and dispensed was charged. There's nothing left in the shop floor. The person said no, anything in the raw material stores, unused material from this particular API. So they went and checked, and luckily for them, there was still 1 drum left. So they decided, fine, let's see. And they sampled it. 1 sample from the bottom, 1 from the middle, and 1 from the top. They have always been sampling this way, except that they always, as seems to be the practice in the industry, of compositing everything. This time they didn't composite it, they ran the 3 samples separately, and they were horrified to find the top layer was assaying at 100%, the middle layer was assaying at 60%, and the bottom layer was assaying at 30%. To make sure, they ran the test all over again. To make doubly sure, they went and sampled all over again, and the same results. Now they composited the samples and ran it, and they got an assay of 65%. Now they knew where the problem was. This is exactly what I meant earlier when I said that misfortune may be the result of a chain of critical events. But success in failure investigation is possible only when you are able to get back every one of those links in the chain, to form that chain, to trace back. Even if one link is missing, then your failure investigation can halt with no further progress. If this particular drum had been used up in that earlier batch, there would have been no raw material left for them to check. And this failure investigation would have met with a dead end. There was another case where they had a mother liquor prepared in the morning, and part of that was used up. One lot was used in the morning batch, and the second lot was used up in the afternoon batch. There was never ever any problem with the morning batch. But every second or third day, there would be a problem with the batch taken up for processing in the afternoon. The microbial burden went beyond acceptable limits, and this happened consistently. So an investigation was launched. The mother liquor had lactobacillus. There was no proper study undertaken to determine the storage conditions appropriate for the material which is being held till usage in the afternoon. There was no study about the time limit up to which this mother liquor could be held. And what was happening was the lactobacillus was multiplying, especially in the summer and monsoon months. There's another instance where again there would be no problems with the first 4 batches, and the 5th batch would suddenly show tremendous amount of microbial contamination. They experienced it once, they experienced it twice, and each time they had this problem, they just take a particular tank which was causing the problem and completely thoroughly sanitize it and take it up for production. There'd be no problem for the next 4 batches. Again, the 5th batch or the 6th batch would fail. When this happened repeatedly, they decided to institute an investigation. And it took them quite a while to try and find out where this contamination could possibly come from. They looked at all possible sources. They identified one small, innocuous little protrusion, which they suspected might be serving as a dead leg and seeding the tank. So this time, as soon as the batch was completed, before it could be taken up for cleaning, they decided to swab that particular area and check for its bioburden as against other parts of the equipment and the bioburden. And they found that there was, as they had suspected, that there was enormous concentration of microorganisms in that little part which had hitherto gone unnoticed. So what was really happening was, even after they were cleaning after the batch, the water remained in that little niche, and then there was microbial proliferation there, which then transferred to the contents in the tank. In another environment, which is a grade A environment, an aseptic processing area, there was suddenly Pseudomonas contamination. They couldn't possibly understand where Pseudomonas could have come from. They checked the HEPA filters, they checked the environmental monitoring records, they checked swabs from various surfaces, but they were making no progress. But the batch remained contaminated. Till one microbiologist said that, listen, since Pseudomonas is basically a soil-borne bacteria, it could be, the only way it could have reached the sterile area is through somebody's feet. So they decided to swab all operators' soles of their feet. And then they identified that operator who was having some infection on the sole of his feet. They isolated him, treated him, etc. But from that day, swabbing of operators' soles has been instituted as CAPA for this particular manufacturing organization. Fine. Now let us say we have done the brainstorming. We have tried to speculate on what could have possibly caused the failure investigation. what could be the cause and what could be the link to the final failure. Now, how do we proceed from here? Hypothesis development. Linking analysis data to causal factors. In causal analysis, identify and list most likely causes, the top 3 to 5 causes that you have all individually identified. Develop utilizing a rating scale or a rating scheme, has its evaluation or risk assessment from high, moderately high, moderate, moderately low, low, whatever, 1, 2, 3, 4, 5, whatever be the way in which you are assessing that risk. Be specific so that the contribution of each cause can be evaluated individually. Now you go into the development of the hypothesis, develop one or more scenarios to describe the failure based on the failure investigation data and the causal analysis. You must be able to evaluate this hypothesis as well as test this hypothesis. Now that you have several hypotheses ready, now the time for testing them. Data analysis, appropriate statistical method. Does the data support the hypothesis? Yes, continue to corrective or preventative action. But if it is no, you may have to revise the hypothesis or causal analysis. And if it is neither yes nor no, but it is maybe, then further experimentation may be necessary. Now, if you have had to revise the hypothesis, now you have to revise the causal analysis based on experimental results, revise hypothesis statement, and go back to retest that same hypothesis to see now whether the data supports this new hypothesis. Now, I mentioned before and I mention it again because it is so important. You must always try to fit the hypothesis to the data, but never try to fit the data to your hypothesis. Now, I also said if you got a gut feeling, trust your gut feeling. Even if everything around you negates that, says it's not possible, you have a gut feeling that this is the reason, go ahead. But don't go so far ahead as to manipulate the data to support your hypothesis. If you look at the data and the data is not supporting your hypothesis, but you're very sure that that must be the reason, then go out and get that additional data, bring it here and establish that your contention was true. The data that you got in front of you, as I said, has got signal, has also got noise. And it may be incomplete. It is your job to go and complete the data that you need before you can arrive at any final conclusion. So you start with the hypothesis and test that hypothesis. Does the data support it? Doesn't support it. Reject that hypothesis. It is supporting, go to CAPA. But if it is, you can neither say yes nor no, then that means you need more additional data, if necessary, even more additional testing to prove your point. Only when your hypotheses are all completed, as I said, there could be more than one root cause. So once you got all that in place, you move to the next stage. And what is that stage? Now the final stage is root cause identification. Summarize results of hypothesis evaluation. Identify all root causes that significantly contribute to failure. Identify significant interactions between root causes. Prioritize root causes based on impact and formally document the results. Every probable root cause is identified and appropriate data collected. The results of the data collected are documented and organized. Everything related to the problem must be identified, but the primary goal must be to find the root cause, the primary cause. The data collected is organized into a usable form. The resulting documentation should address all of the probable root causes previously determined. This information is used to determine the root cause of the problem. The effectiveness of the analysis will depend on the quality and thoroughness of the information available. Now you have an example of a root cause summary table. It is relating to a bag seal failure, and it has listed 4 possible root causes, and one is temperature variation, the other is sealer cycle time, third is the material thickness, and lastly the accuracy of calibration. The temperature and sealer cycle time are high impact, and the material thickness and accuracy of calibration are moderately important. Please remember, what we have now discussed is failure investigation and root cause analysis and identification. I've also said the failure investigations team's core competency is investigation, need not be CAPA. So the failure investigation team tells you what went wrong and how it went wrong and why it went wrong. It need not necessarily give recommendations on CAPA. If it does, it's good. You can have a section which says recommended remedial, corrective and preventative action and list all of them. But if you don't, it's perfectly alright. You're closing your failure investigation file, you've identified the root causes. Now, no failure investigation or root cause analysis is complete without proper documentation. Now, as I said earlier also, the documentation should be done such that somebody else can independently, without referring to you or your team, can understand exactly what had happened, what was investigated, what was the investigation findings, and if there are investigation recommendations, good. But otherwise, there's enough material for the next person to take the ball and run from there to implement CAPA. Now, the documentation could be different in different organizations, but generally the structure is as I am describing now. Key elements of documentation: specific problem statement, the traceability and tracking information, the failures, the test samples, the failure investigation itself, the observational data, the inspection data, the test information. Now, you could have a protocol where you record the data analysis and the results and conclusions. You then go on to causal factor identification, how you went about that process. Hypothesis development and how you went about that process. Hypothesis testing and verification. You may have a protocol for that, again for data analysis, results and conclusions for establishing those hypotheses. And lastly, the root cause summary or the report or the table or the map, whatever form in which you present your final data. Here are some points to consider while you do your documentation. How detailed do I need to be in my documentation? Detailed enough so that someone from the outside can follow your logic and could replicate your results. Detailed enough to meet specific regulatory requirements, that is complaints or adverse reactions or deviations. Do some areas need to be more detailed than others? If so, what areas should I emphasize? The answer is yes, of course. Different areas require different levels of details. The problem description is important, the traceability is important, the failure investigation itself is important, and the root cause summary is critical. Do I have to go through every step for every investigation? No, use common sense and right size the activities for the project. Wherever the term investigation is used in a regulation or a standard, a certain level of rigor is implied. So again, focus on the problem description, the traceability, the investigation, and the root cause summary. For complaints not reportable to FDA, when can I avoid doing all this investigation and root cause analysis? When a similar complaint has been fully investigated, you can refer to such an investigation. When the event, based on documented data analysis, is very obviously an isolated event and does not represent either a trend or a pattern, then you can dispense with failure investigation and root cause analysis. Of course, this does not relieve the requirements for review and evaluation for complaints related to core product requirements. Other points to consider: please remember, whenever in doubt, write it down. If you were an auditor, what would you want to see to ensure activities were done correctly? If CAPA is done by another group or person, what will they need to complete the project? If you were gone, would someone be able to follow your process and reach the same conclusion? Whatever it is, your investigation must be well organized, documented, evidence-based and transparent in order to provide a framework for analysis with the ultimate goal to support decision making regarding CAPA. Here is a quick summary of whatever we have discussed regarding failure investigation and root cause analysis. An overview of the RCA process. Root cause analysis is a generic term used commonly to refer to structured problem solving within organizations. Root cause analysis is a step-by-step method that leads to the discovery of a fault's first or root cause. Every failure happens for a number of reasons. There is a definite progression of action and consequences that lead to a failure. An RCA investigation traces the cause and effect trail from the end failure back to the root cause, much like a detective solving a crime. The method of root cause analysis must be determined on a case-by-case basis. Normally, it will be construction of a fault tree analysis or causal tree analysis using the why methods of investigation. It may be inductive or deductive. It may also utilize Ishikawa fishbone analysis. It shall be linked to risk analysis procedures to determine priorities once causal analysis is completed. The method brings a team of 3 to 6 knowledgeable people together to investigate the failure using evidence left behind from the fault. The team brainstorms to find as many causes of the fault as possible. By using what evidence remained after the fault and through discussions with people involved in the incident, all the non-contributing causes are removed and the contributing causes retained. A fault tree is constructed, starting with the final failure and progressively tracing each cause that led to the previous cause. This continues till the trail can be tracked back no further. Each result of a cause must clearly flow from its predecessor, the one before it. If it is clear that a step is missing between causes, it is added in and evidence looked for to support its presence. Once the fault tree is completed and checked for logical flow, the team then determines what changes to make to prevent the sequence of causes and consequences from occurring again. Now that we have completed failure investigation and the root cause analysis, we are now ready to proceed to the next logical step, and that is to bring about corrective and preventative action. So before we discuss that any further, let's understand the definitions. Corrective action is the action taken to eliminate the cause of an existing nonconformity, defect, or other undesirable situation in order to prevent recurrence. Preventative action is the action taken to eliminate the cause of a potential nonconformity, defect, or other undesirable situation in order to prevent occurrence in the first place. I said the failure investigation team should be a crack team of experts with certain attributes. And I also said it is not necessary that the failure investigation team should be the same team that also does CAPA. CAPA is also a specialist job, and you could have a totally different team executing CAPA. Planning the CAPA, executing the CAPA, monitoring the CAPA, reviewing and closing the corrective and preventative action. Now it could be possible that some members of the failure investigation team also double up as members of the CAPA team, but that is on a case-to-case basis and situation basis. I had said that for failure investigation, the trigger could come from internally, from within the organization, like a batch failure, like a serious OOS, serious deviation, or a stability problem. Or the trigger could come from outside, from the market. A customer rejects your batch, or there are complaints from a customer, or there's an adverse event in the field. In the case of CAPA, it goes way beyond the failure investigation trigger. There are far many more triggers for CAPA. One, of course, is the result of failure investigation. If there is a change in the regulatory environment or regulatory expectations, that triggers a CAPA. For example, until now you were working in an unclassified environment and the regulations have changed and now they say that you shall work in a certain grade D environment or a grade C environment. It triggers a CAPA. Now, it could also be because of regulators coming in for inspection and giving you a long list of non-conformances, critical deviations, or critical defects, critical deficiencies, or major deficiencies, or minor deficiencies. And if these come to you, they come to you with a certain time frame within which you have to commit to execute your CAPA and be ready for a follow-up inspection. The trigger could come from inside, like an internal audit, or an annual product review, or management review. Just around anywhere, the internal or external. The trigger triggers a CAPA. And it is now for the CAPA team to get its act together. So what should it do? Now, just as in failure investigation, you have to submit a proposal to management and get their written approval to initiate the failure investigation, so too in CAPA. You make a proposal of what you intend to do and why it is important, backed by the failure investigation reports or whatever be the trigger that has triggered this CAPA, and obtain in writing assent from management that you may start your CAPA. Another important thing to do before we get started on CAPA is to find out how much support are we going to get from our management. The regulators now expect that management will lead. So management has to be part of the CAPA team in terms of CAPA initiation, CAPA monitoring, CAPA review. So how much support can we expect from management? Next, what is the enforcement acceleration rate? For example, if it is a regulatory non-conformance, you have a window within which you have to complete your CAPA. So how quickly should this CAPA be taken up? What is the priority to be given to this project? Next, how should the process be managed? Can we do it internally, or should we appoint a professional project manager to get the job done? And lastly, do we need consultants to complete this job? If so, how should these consultants be used? Now that you've got that assent, don't just jump into CAPA. Just stop, pause, and take a look around. You have the root cause with you. Determine the extent of the problem. You got an isolated tank. How many more such tanks do you have in the plant? How extensive is the problem? For example, not only in your plant. If you are a multi-locational facility, the same thing could be there in your other facilities. Now it's surprising that you get a complaint from a customer. That complaint is not circulated to all the plants. If the complaints are circulated to everybody, and if the complaint is being addressed in one location, the same could also be checked in the other locations and the same CAPA could apply there. When an inspector inspects one facility and gives a list of non-conformances, that is not circulated to all the other locations to check whether they too have these same deficiencies that they could be covered under the same CAPA. Now you have an internal audit in one facility. Why don't you circulate that to all the facilities and find out these are the deficiencies in this facility, are there similar deficiencies in other facilities? So effectively it becomes very important to try before you commence your CAPA to find out the extent of the problem and the extensiveness of the various problems. Don't just look at this particular fault, this particular root cause analysis and initiate a CAPA. Review to find out how many other things are hanging fire which have not yet been resolved, and try and see if you can include all that in one single CAPA. Prevent recurrence at a system level and prevent recurrence at a corporate level. So what does this really mean? It says, don't just look at it in that narrow viewpoint of a failure and a root cause and redress, but look at it as a system. Is there a policy in place? Can we back this by procedures? What all needs to be done at our level, in our plant, in our organization? And can we then replicate this at a corporate level so that all locations benefit from the one incident? Please remember that I had said earlier, accidents don't just happen. You get a series of warning bells. Now, this failure might be just a small signal to you that things are not quite under control in your plant. For example, you landed up producing the wrong product altogether. Everybody was looking, but nobody was seeing. Is this a phenomenon in all the other locations as well? So, whatever preventative action you're trying to take, make sure it's at a system level, at the top level, backed by procedures, and also at a corporate level. Now, when we are planning CAPA, we are trying our best to eliminate the root cause, or at least minimize the probability of failure through that root cause. Now, whether you're going to do risk elimination or risk reduction, the preferred hierarchy of strategies is always eliminate or reduce or mitigate by design. And if that is not possible, eliminate or mitigate by automation. And if that also is not possible, then you resort to elimination or minimizing by systemic control, procedural control. If it is something as simple as a tank with a protrusion that was seeding the batch and contaminating the batch, and all you had to do was get rid of that protrusion, and you eliminated the problem once and for all, it's excellent. But such solutions are not always possible. You had a Pseudomonas contamination as part of your CAPA. You said in future all operators' souls shall be swabbed as part of the profiling. It's not always possible to eliminate the cause. For example, if it is a human error, for as long as you have a human there, you're bound to have an error tomorrow or day after, at one time or the other. So you will have to look at other ways of either eliminating the risk or minimizing the risk of the next failure. People who had this problem with glycerol monostearate and glycerol distearate went to the manufacturer and said, if both are amorphous powders, it will be very difficult for our people to distinguish it. So I want some visual difference in these two materials. So they got GMS as flakes and GDS as crystals, granules. So that even if there was a mix-up in processing, hoping that people who are looking or also seeing will know that when GDS is being prescribed, the material is appearing in flakes, it cannot possibly be GDS, it must be GMS, and the process halts right there. Now, if you cannot eliminate by design like this was a case of elimination of the risk or the root cause by design. You go to the next step, which is elimination or risk reduction by way of automation. So if here there is somebody who is weighing 12.1, charging 12.1 and writing down 11.2, which is what the BMR has asked. If you have a weighing machine with a printout, it will tell you at what time what amount was weighed and charged and what is the order in which materials had been charged. For example, 11.2 or 11 kgs followed by 13 kgs followed by 4 kgs. The printout will show whether 11 kgs plus 13 kgs plus 4 kgs were indeed charged in that order and that weight. Now, it will not be able to tell you whether it was 11 kgs of A or 11 kgs of B. Now, if you can make that arrangement as well, that's good for you. But at least you have taken this much care. You have now been able to support what the BMR is saying, what the person is recorded as having done, backed by a printout from a machine saying what was really weighed at that particular point in time. In the case of the lactobacillus, a decision was taken that the mother liquor would be prepared in the morning for the morning batch, and mother liquor would be prepared separately in the afternoon for the afternoon batch and taken into production immediately without any kind of lag time in between. In the case of the raw material that had assayed at different values at the top, middle and bottom, an SOP was rewritten to say that material from this supplier at least will always be sampled at 3 different levels and tested separately and not composited. So these are some of the examples of risk elimination or risk reduction by design or by automation or finally by procedural controls. Now let's look at how we proceed with implementation of CAPA and what all we should do and what all is expected of us. Preventing recurrence of the failure. Once the fault tree is completed and checked for logical flow, the team then determines what changes to make to prevent the sequence of causes and consequences from occurring again. It is not necessary to prevent the first or root cause from happening. It is merely necessary to break the chain of events at any point and the final failure cannot occur. Often the fault tree leads to an initial design problem. In such a case, redesign becomes necessary. Where the fault tree leads back to a failure of procedures, it is necessary to either address the procedural weakness or to install a method to protect against the damage caused by the procedural failure. Using the results of failure investigation and root cause analysis exercise, the best methods for correcting the situation or preventing a future occurrence is determined. All of the tasks required to correct the problem and prevent a recurrence are identified and incorporated into an action plan. The plan includes changes that must be made and assigns responsibility for the tasks. Employees shall be trained and all the affected functions shall be communicated. Now, actions to be completed: list all activities and tasks that must be accomplished to correct the existing problem or eliminate a potential problem and prevent a recurrence. It is very important to identify all actions necessary to address everything that contributed to or resulted from the situation. Now, changes. Needed changes to documents, processes, procedures, or other system modifications should be described. Enough detail must be included so that it is clearly understood by everybody as to what must be done and what the outcome of the changes ought to be. Training and communication. Employee training is an essential part of any change that is made and should be made part of the action plan. To be effective, all modifications and changes made must be communicated to all persons, departments, suppliers, etc. that were or will be affected. Next is the implementation and final closure of CAPA. The action plan that has been developed is executed and all identified tasks and activities completed. The actions that were taken are summarized and all modifications to documents, processes, etc. are listed and recorded. Finally, effectiveness and monitoring. One of the most fundamental steps in the CAPA process is completing an evaluation of the actions that were taken. The evaluation must not only verify the successful completion of the identified tasks, but also assess the appropriateness and effectiveness of the actions taken. Now, it helps to have a checklist or series of checklists. Check if all the objectives have been met. Did the actions correct or prevent the problem with assurances that the same situation will not happen again? Have all recommended changes been completed and verified? Has training and appropriate communications been implemented to assure that all relevant employees understand the situation and the changes that have been made? Has an investigation demonstrated that the actions taken have not had any additional adverse effect on the product or service? Next is monitoring. Make sure that appropriate information has been recorded that provides proof that all actions have been completed successfully. And next is validation. A validation of the action is done. This must document that the root cause of the problem has been solved, any resulting secondary situations have been corrected, proper controls have been established to prevent a future occurrence, the actions taken had no other adverse effects, adequate monitoring of the situation is in place. And finally, closure. When the follow-up has been finished, the CAPA is completed and closed, duly documented by quality assurance. And finally, I'm placing before you the thoughts and recommendations of an author who talks about CAPA implementation in 4 phases. In the first phase, it's all about preparation. In the second phase, it's all about design. In the third phase, it's all about execution. And in the last phase, it's all about verification. Now, what are the recommended activities at each of these phases? Let's take a detailed look. 4 phases and 20 tasks for an effective CAPA plan. For each of these tasks, you have a start date and end date, and you have a start date as per the plan and the actual start date, and the end date as per the plan and the actual end date. In phase 1, there are 5 steps. Form and train a team, research, analyze issues and develop as-is process flow diagram, develop project scope, alternatives, assumptions, obstacles and cost, develop communication plan, and now review this phase of preparation and make a presentation to management. Once this is accepted by management, you move to the design phase. And here again, there are 5 specific steps. First, develop the to-be process flow diagram. Review existing policies and procedures, develop new policies, procedures and architecture, confirm the design with subject matter experts. Now, once again, you review this phase of preparation and make a presentation and obtain approval from management. You are now ready for the next phase, which is implementation, which is again in 5 steps. Obtain approvals of new procedures, develop implementation plan, process through change control and documentation control system, conduct general awareness and specific job training. And at the end of this, review this phase of internal audit, that system is in place and in use. And lastly, you move on to the verification phase, where you assemble verification documents, conduct internal review and approvals of verification document, issue and review verification document with third-party reviewers if necessary, follow up on outstanding issues identified. And last step is the conclusion, file the project documents and schedule internal audits to check for continued compliance. Now, this author also goes on to speak about why CAPA seldom works, why CAPA so often fails. Now, he has listed 5 reasons. Why CAPA fails? Failure to analyze, failure to agree, failure to communicate, failure to execute, or most often, failure to follow up and verify satisfactory implementation. I hope you found this presentation useful and interesting, and thank you for your attention and for your time. Thank you very much.